Okay, our next speaker is Meera Heller from UC Davis in California, and she's going to talk about sodium iodide inactivating two bugs in vitro.
Yes. Thank you, and thank you for the opportunity to speak here this morning. I'm going to talk about sodium iodide and its in vitro inactivation of some of our common bovine respiratory pathogens. So I don't think I need to tell people in this room how devastating bovine respiratory disease is to the cattle population and the pocketbooks of our clients. And also, we've been hearing a lot about the use of antimicrobials, and it is, as we know, a major reason for use of antimicrobials in the bovine industry. So it's a multifactorial disease, and current dogma sort of says that stress, such as weaning stress or transport stress, coupled with a viral infection, then decrease the immune defenses and allow bacterial pathogens access to the lower airways and cause pneumonia and disease. And our control programs so far center around the use of vaccines and also widespread antimicrobial use, both as metaphylaxis and as treatment. So I'm going to take you back in time to think about sodium iodide again. And historically, in the pre-antibiotic era, it was used for a wide range of diseases, fungal diseases, bacterial granulomatous infections, and most notably tuberculosis in humans. And this is a little beautiful little pipe that was used to inhale sodium iodide for treatment of tuberculosis in the 1800s. So it was fairly widespread use in humans. Currently, we bovine veterinarians use it for actinobacillus and actinomyces infections in cattle. But despite this long time use of this substance, we still don't really know what its mechanism of action is. One possibility is that it could be a substrate for peroxidases. And peroxidases exist in our white blood cells, such as phagocytes, and they also are present on our epithelial surfaces. So lactoperoxidase is a peroxidase that was first found in the mammary gland, hence the name lactoperoxidase, but it also is in the saliva and in airway secretions. And it catalyzes a reaction between hydrogen peroxide and a halide to form a toxic hypohalous acid. And in our normal body system, in you and me, our body creates a pseudohalide called thiocyanate, which is transported out onto the epithelial surfaces of your body and acts as a substrate for this enzyme. And this product is mildly antibacterial. However, if there is sufficient iodine present in the body will replace this thiocyanate with iodine and transport it out onto the epithelial surfaces. And when iodine is used as the substrate for lactoperoxidase and forms a hypoiodous acid, this product is antibacterial and antiviral. And this was shown by Fisher in 2011, published a paper that looked at sodium iodide in this system and its effect on adenovirus and human respiratory syncytial virus. And you can see, if I can make this work, that in the cell cultures that used sodium iodide at two different concentrations, so at high concentration and low concentration, there's no viable virus left. They went on to show that they could deliver a single dose orally in humans and achieve significant increases in airway iodine levels in the airway secretions that were equivalent to what was necessary in vitro for killing. And they went on to do a challenge study using human respiratory syncytial virus in sheep and show that there was protection in vivo in that challenge. So looking at this data, we were thinking, can this be applied to bovine respiratory disease? And this would be a great way to sort of increase the activity of this already present innate antiviral, antibacterial mechanism that we know is present in cattle. And in part, we know that because this system is of interest for cystic fibrosis research. And the early models for cystic fibrosis research were bovine tracheal explants from slaughterhouses. So we didn't have to do the work to show that it's present in cattle, which was nice. So the current knowledge gaps that we have are, is this system effective against our bovine respiratory pathogens? And then secondly, can we deliver this orally and increase respiratory secretions? So we obtained some clinical isolates of Manheimia hemolytica and Bibersteinia trehalosi from bovine pneumonia cases at necropsy that were submitted to our state lab. And this is in vitro. We used varying concentrations of sodium iodide from no sodium iodide to 500 micromolar sodium iodide with the reaction mixture, so with the lactoperoxidase and the hydrogen peroxide. And samples were taken prior to the addition of hydrogen peroxide because the hydrogen peroxide is the catalyst for this reaction. And then samples were taken at both 5 and 30 minutes of the reaction. So I sort of think that a picture is worth 1,000 words. So this is an example of one of our plates, and we saw fairly consistent results across the different strains of each bacteria that we tried, that the complete reaction mixture of the lactoperoxidase, sodium iodide, and hydrogen peroxide inactivated both of our bacterial pathogens at all the concentrations tested. So you can see from this plate we have growth in our 0 concentration of sodium iodide, and then no growth in all of our experimental concentrations. And then these are all of our control situations where we have some growth. And this is a little bit more scientific representation of the data. And the blue bars are the varying concentrations of sodium iodide in the complete reaction mixture, and then all the other colored bars are our control conditions. And you can see that there's, even after 5 minutes for the Bibersteinia trehalosi, there is no growth in any of our sodium iodide concentrations. And there we go. Results were similar for the Manheimia hemolytica. Although at the lowest concentration, we still had some growth of Manheimia at the first time period. So there might be a little bit of a time-dependent and concentration-dependent killing for this. So we also wanted to see if this worked for our respiratory viruses. So this work was done by B.J. Newcomer at Auburn University, and he used three of our bovine respiratory viruses, bovine herpes virus 1, parainfluenza 3, and bovine diarrhea virus, and cytopathic strains of each virus were used. And this experimental conditions were very similar to what we used for the bacteria, although I will just point out that the concentrations of sodium iodide were slightly different due to a slight miscommunication between him and I when we were setting up these experiments. So it's not as pretty as I would like it to be. And virus titration was performed by direct visualization of cytopathic effect on MDBK cells. So these are the results for bovine herpes virus 1, and they're really encouraging. They show dose-dependent inhibition that was at work as early as 5 minutes into the reaction. And the results for parainfluenza 3 are very similar. They show a steady drop in viral titers with a dose-dependent inhibition as well. So that was very exciting for us. However, bovine diarrhea virus unfortunately seems to be not susceptible to this innate immune mechanism system. So we were a little bit sad about that. So our next question was, can we replicate the human study where they were able to feed humans, or I guess have humans take sodium iodide and increase airway secretions of iodine? So this was done by Dr. Brian Vanderlei and Dr. Brian Shoemake at University of Missouri, and they used 16 weaned commercial crossbred beef calves and basically dosed them one time orally with 70 milligrams per kilogram of sodium iodide, and they did a crossover design where some of the calves received water as well as an equivalent volume. And they collected samples of nasal secretions using this very ingenious little basically nose vacuum, using a serologic pipette and a vacutainer tube and then a vacuum source to vacuum out nasal secretions. They collected nasal secretions and serum every 12 hours for 72 hours. And you can see the results from the nasal secretions. They achieved high levels of sodium iodide, or just iodine, sorry, in nasal secretions. At the first time point, they were already quite high. And I've marked on here the 500 micromolar and 250 micromolar, which were what our in vitro concentrations were, just so you can compare. But you can see that out till 72 hours, we are able to have airway secretion levels that are equivalent to what was effective in vitro for at least the pathogens that we looked at. So I think we can say that in vitro, at least, this looks really promising. It inhibits IBR, PI3, Manheimia, and Bibersteinia. And also an important point is that these concentrations were not toxic to our cultured cells, so that's another important point to make. We didn't look at BRSV, but we think that it's likely that it's susceptible as well based on the work that was done previously in humans and sheep. And unfortunately, BVD was not susceptible. I think it's important to remember that BRD is a multi-pathogen disease, so there's still more pathogens out there. We don't know if all the pathogens will be equally affected. I'm getting ready to try mycoplasma as soon as I get back home. And I think as we learn more and more about the microbiome, I wonder if we do these things in vitro, and if we're going to end up just manipulating or changing the flora in the nasal passage, and maybe we'll see some other pathogen emerging and not actually decrease disease. So that's another caveat to think about. And then future questions are, can we reduce respiratory disease by strategically administering this on farm? And then the holy grail would be, can we reduce antimicrobial usage on the farm using techniques like this? So just like to thank all of my co-authors. This was a team project, and hopefully answer any questions that you may have.
Okay. Okay. Any questions from the floor? Maybe I could ask a strange question. Would iodide change the microbiome in the gut if you're giving it orally?
That is a really good question, and we actually collected fecal samples. I did a separate study in pre-weaned calves to look at the pharmacology of it, and we took some fecal samples, so we're probably going to submit those and run to see if the levels of excretion, and I think that would be the first place to start. But we're wondering if there's potentially some excretion in the gut, and if it is effective against GI pathogens.
So it's probably absorbed high up in the gut with transporter.
Yeah, that's what I think, because we got fairly high. I didn't show the serum levels, but the serum levels peak fairly high. Okay. And so I'm thinking a lot of it gets absorbed, but I don't know the question about that yet.
Any other questions? Go ahead.
Good morning.
Good morning.
Congratulations for your presentation. I'm Chris from Greece. I'd like to ask you if you have thought how the iodine sodium could affect the function of thyroidal gland and the thyroid hormone, because we have such issues in Greece because of the iodine consumed from the small ruminants sometimes.
Oh, okay.
So you could thought about it, if you have thought about it.
Yeah, I have thought about it, but I honestly didn't think that the dosing, I think you have to be fairly chronic, right, to have those problems. I've never seen them clinically, so I don't know. But we were thinking more this would be a sort of a timed, specific intervention rather than something that would be, fed, because there's also meat quality issues and those sort of residue issues that we didn't want to get into as well. So, yeah, but that's a very good question. Yeah, thank you.
You mentioned toxicology, that there was no toxicity on the cells. So that's MTT assays and things like that, is it?
No, just the cell for the viral titers, so the Madden-Darby kidney.
Okay, MDCKs.
Cells, yeah.
So how are you showing that the iodide is not toxic to those cells?
So we had our negative controls, and they had the cells were still alive.
So it's a viability assay.
Yeah.
Okay.
Yeah.
Right. Okay. With that, we'll move on.
Thank you.
Thank you.